안녕하세요. 오늘 웨비나 진행을 맡은 BMS 김승현입니다. 우선 귀한 시간을 내어주셔서 감사드립니다. 발표 진행에 앞서 몇 가지 안내 사항을 말씀드립니다. 웨비나는 발표 시간 40분, Q&A 시간 10분으로 진행되며 상황에 따라 시간이 조정될 수 있습니다. 질문이 있으신 경우 화면 하단 컨트롤에 위치한 Q&A를 클릭하고 Ask a Question을 클릭하신 후 질문을 남겨주시면 Q&A 시간에 답변 드릴 예정입니다. 질문 언어는 영어, 한국어 모두 가능합니다. 한국어로 남겨주시면 저희 측에서 발표자에게 영어로 전달할 예정입니다. 발표 시작 전 설문 이벤트에 대해 안내드리겠습니다. Q&A 시간이 종료되면 채팅창이나 화면에 설문조사를 위한 링크와 QR코드가 게시됩니다. 설문에 참여하신 모든 분들께는 발표 영상의 다시 보기 영상을 2주 동안 제공해드립니다. 또한 설문에 참여해 주시는 분들 중 30분을 추첨하여 스타벅스 아메리카노 쿠폰을 드립니다. 커피 쿠폰 당첨자는 6월 26일 수요일에 발송되는 다시 보기 영상 안내 메일에서 안내드리며 7월 3일에 발송드릴 예정입니다. 많은 참여 부탁드립니다. 발표에 앞서 오늘 웨비나의 연자 시핑리우 박사님과 BGI 및 스토믹스에 대해서 간략히 소개드리도록 하겠습니다. 시핑 리우 박사님은 BGI 리서치의 생물정보학, 종양학, 뇌과학 분야에서 선임 연구 교수로 재직 중이십니다. 지난 5년 동안 30편 이상의 SCI 논문을 발표하며 다량의 학술 성과를 거두셨습니다. 특히 싱글셀과 트랜스크립토믹스를 활용한 연구 주제로 네이처, 사이언스, 셀과 같은 저명한 저널에 9편의 논문을 발표하셨습니다. 이번 웨비나에서는 단일세포 공간전사체학을 활용한 뇌과학 분야의 응용 사례를 다루며 공간전사체학의 실용적인 활용 방안에 대해 통찰을 제공해드릴 예정이므로 공간전사체학의 최신 연구 동향과 실제 적용 사례를 얻어가실 수 있을 것으로 생각됩니다. 연자가 속해 있는 BGI는 1999년에 설립된 세계에서 가장 큰 유전체학 연구기관 중 하나입니다. BGI는 유전체학, 생물정보학, 분자생물학 등 다양한 분야에서 탁월한 연구 성과를 거두고 있으며 전 세계 100개 이상의 국가 및 지역에서 장비, 기술 지원 및 솔루션을 제공하고 있습니다. BGI의 자회사 중 하나인 스토믹스는 BGI의 축적된 유전체학 노하우와 자원을 이용하여 공간 전사체학 기술을 선도하는 기업으로 유전자 발현의 공간적 패턴을 단일 세포 수준에서 고해상도로 시각화하고 분석할 수 있는 혁신적인 기술을 제공합니다. 오늘 웨비나를 통해 BGI와 스토믹스가 제공하는 스테레오섹의 첨단 기술과 서비스를 살펴보시면서 여러분의 연구에 도움이 되는 방안을 찾으실 수 있으시기를 바라겠습니다. 다시 한번 참석해 주셔서 감사드리며 오늘의 웨비나를 시작하겠습니다. Dr. Liu, could you please put up the slides and begin the presentation?
Okay, thank you for the introduction. I will share my screen. Okay, could you see my screen?
Yes, we can see.
Okay, thank you. Thanks for the introduction by Dr. King, and it's my great honor to be here to introduce our latest advance in omics technology and its application on the study in the brain science. And I come from BGI research, and my research interesting is focused on the big data and also its application in the tumor and the brain science. So today, my speak title with the cortical evolution in amniotes, insight from a comparative brain atlas. As you can image that we used the single cell and spatial sequencing technology on the field of the evolution research, which is a popular field that traditionally uses the DNA sequencing research. So we know that the major scientific discovery and the new technologies often propose each other. For example, in the 17th century, and scientists like Galilei improved the invention of the microscope and enabled the robot hook to observe the structure of the cell in plant samples using the microscopes. So that's opened a new chapter in the cell biology. In the last century, the invention of the DNA sequencing technology in the 1977, especially with the complete of the Human Genome Project, opened a new chapter in omics research, opened the era of the big data research in the life science. So the recently emergence of the spatial omics technology, we believe, will be another breakthrough, such as the spatial transcriptors technologies developed by BGI in 2013. We called Stereo-seq, which has pushed the omics research from the DNA molecular and the single-cell level to the analysis of the structure and the function, and we believe that as related technology development, the future research in the life science will enter the fast track. So today, BGI's omics technology has achieved a sequencing from the DNA to single-cell level to spatial omics. We call it the DCS levels, which include the sequencing technology and the DNA, cell, and the spatial levels. Based on the DNA DCS level, we can study the fundamental laws of the life science from different aspects, such as analyzing the basic structure of the brain and ultimately understanding high-level cognitive function like causal science. Study the origin and the evolution of the species from a completely new perspective, and understand how life developed and from a single egg to a complex individual with tens of billions of cells, and in the end, researching disease mechanism to achieve the long-term goal of the precision medicines. So I believe the DNA sequencing technology is already well known to everyone. So since we released the first BGI sequencing about 10 years ago, our BGI teams have updated and replaced it with new generations, achieving different applications, including the delivered with the different sequencing output, such as the G400 and the T7, and now we come to the T20. So in the terms of the single-cell technologies, we also have developed independent automated cell sorting and library building systems. We named the DNB-enabled C4, and we now we come to the type M. So moreover, MGI have already achieved our complete upstream and downstream systems, enable delivers from the sample storage and the transport to the end for the data analysis. So using the DNB-enabled system, we achieved the first ever sequencing of the body atlas from monkeys. We sequenced over 1 million cells from 45 minor organs or tissues across the body. And in the end, we are achieving about leave of our counselors in mapping the body's cell landscapes. Specifically, we identified 106 main cell types, and among of them, and 463 cell subtypes. So this study has published in Nature in 2022. However, single-cell technology can only tell us about the composition of the cell type, but like the part of the car, even if we know every component, it's still difficult for an ordinary person to understanding the structure and the function of a car. So let alone assembly and improve it. The breakthrough in spatial omics technology have made up for the limitation of single-cell technologies, allowing us to obtains spatial location information for its molecular and its cells. So this enable us to understand the structure of living tissues, just like the two alternates below. So the left one shows the single cell clustering result. Every single color represents different cell types, and in the in the right is also the spatial technology dataset. So this is very similar to a map, but is already totally different. We can clearly see the details explanation of its genes in itself with the spatial information. I believe this information will be shocking and exciting for every smart scientist. So in the 4 years ago, and based on our DNB nanobot sequencing chip, we have developed the Stereo-seq. We also named the ST-omics, which is a spatially resolved transcriptional technology that combines billions of the tightly packed barcode in low position and large capsule areas. This is the workflow of the Stereo-seq. We firstly use the DNA nanobot contains randomly barcode sequencing onto a modified chip with a ground patterns array of spots where the DNB are effectively docked in. Each spot being about 220 nanometers in diameters, and with a center-to-center distance of 500 nanometers. So then the molecular identifies we call it the MID, and the protein sequence contains on the oligos or ligand on each spot slow the CID. So computer analysis with this data and allow us to see the whole picture in the 6 steps. So the advantage of this technology is that each cell is about 10 micrometers in diameters, which can cover about 400 dB spots. So this allows the resolution at subcellular levels. In addition, and we can see the size of the chip is available for the up to the biggest chip can be up to about 10 centimeters plus 10 centimeters. So it's allowed to profiling for the large tissues such as the human brain, even for the human brain samples. So using this Stereo-seq, we performed it to the development research of the mouse embryos from the embryo the day 19, the 9 and the day 16. So in the subsequent period of the 16th day, we continued sections sorting slides on the list constructed the 3 dimensions of the embryo. From these amazing results, we can see very clearly for the development process of each tissue and organs. So this article was published as a cover story in the Cell magazines in 2022. So here we also used the Stereo-seq technology to study the brain regeneration after enjoyed in a species we named the salamander, so called the axolotls. So salamander is a very interesting species that it has a strong ability to regeneration. Even the brain have been damaged, it can be repaired in the futures. So we can see that after enjoys in the day 2 is the right side of the brains, but the left side we just take as our controls. So in the second days we can see this is a damage, and also the cell around the damage have been changed a lot until the day 13 and 16, almost recovering to the pre-injury stations. So using the Stereo-seq, we replayed the entire process, especially achieving a single cell level resolutions. So we can see in this picture, and every single spot represents our single cells, and the different color represents the different cell types. So we can see very clearly for in the single cell level, and this paper is also published as the cover story in the Science magazines in 2 years ago. So in the 2022, in the 2 years ago, and we besides the embryo, mouse embryo, and also the axolotl, and we also published some more people about the development and for the Atlas, including the 4 papers in the Cell series and 2 papers in the Nature, and 1 paper in the Science. So 5 of them have been as the cover story published. So under the question we want to ask, besides the DNA sequence and the single cell sequence, so what can we do with additional spatial information? So as you can see, the Stereo-seq can get the spatial information. So we summarized our answers into the 4 dimensions. For the first of all, and is the 0 dimensions we code. So even because we know there are many different methods about the spatial transcriptor, and every technology should be do the cell segmentation, because the cell level is everyone want to get the data. So for the high resolutions, we need to do the cell segmentation. But for the low resolutions, we also need to do the deconvolutions. So in this first dimensions, so we could do the axolotl genes on the 1-dimension passes, and to the 2 dimensions, we could do the spatial domain identifications with the under is the spatial variety genes and the cell-to-cell communications. And for the 3 dimensions, we can see the mouse embryo in the 16 days. So we can reconstruct the 3 dimensions of the whole tissues and even for the whole embryos. And in the 4 dimensions, we could see a series of the time points in the samples. For example, for the cell migration path over the times and the mechanism, and also if we comparative across the species, actually we could do the evolution of some of the cell type or some of the domains. So in the end, we also developed our toolkit, like we named the spatials, and the spatials can deal with most of the single cell and the spatial transcriptional data with a very amazing functions. For example, we can do the single cell cell segmentations based on the image and based on the RNA signal enrichment. And then we also can do the domain identification and digital laser. And also it's amazing that we can do the cell-to-cell communication in under spatial niche, but it's very different between the single cell-to-cell communication, because the single cell communication is based on the receptor and the cell type based on the base. For example, for the cell phone DBs, but for the spatial ones, and we can see the physical location to see the cell around the cells into for the physical communications to each other. And also in the toolkit, we also have a function to do the 3-dimensional reconstruction and the models. And in the last also we can do we use our vector field and the model to see the cell migration and also the cell development. Also, the toolkit have been published in the preprint in the backup, and also the paper have been submitted to the cell for the reviewing. So we come to the blind science in BGI, and we know the blind science is very popular in the last study. And but in BGI, actually, we only focus on we use the our latest omics technology. For example, for the DCS label technology I mentioned before. So we use the DCS label technology to the four directory of the brain science. The first is Atlas. So we want to build the modern animals brains, for example, for the human and the mouse and also the macaque and also the fruit fly and so on. And then we use the because after we get the different species the Atlas, we could do the evolution of the brains. So and then we also have interested in the disease of the brains. For example, for the Alzheimer and Parkinson disease, also the deplacing for the youngers. And in the end, we also focus on our brain science in the interface. So we want to use all the data we get based on the Atlas evolution and the disease. We want to developing a next generation of the interface based on the big data. And also including the AI advances. So the human brain is actually an organ with very complex structure and very powerful functions. So because of this, resource on the brain appears to be very challenging. It's extremely important. We need to study the brain from a multiple level, from the cells and from the cell-to-cell linkers, and also the interactions. For just like our science review on the 2021 by Dr. Li Qunlou, it contains from the letters to word and to sentence in the end to the article. So for an articles, of course, there are keywords and the sentence. However, for complete understanding of our articles, it seems that missing any part will be result in the loss of some information. So it seems that all the contents appears to be very important. So it mentioned that the cell type and the structure, actually we also have the technology to do the resource based on the cell type. The cell type is our single cell omics technology, and structurally we could use the ST omics technologies. So starting from the resource of brain structure and cell type and multiple levels, we cooperated with the scientists from the Institute of the Neuroscience based in Shanghai, including Mu Minpu and Chen Yunli and Yan Gangsun's teams. We reconstructed the three-dimensional gene expression map of several model animals' brains, including the mouse and marmoset and the macaques, taking the macaque brain as the case. So we section the brain from the anteriors to the posteriors with an interval of about 500 the micrometers. So resulting in over more than 100 whole brain cell slices for Stereo-seq, covering an area of up to 5 centimeters plus 4 centimeters. For the remaining tissues, we also sampled brain regions and performed in the single cell sequencing, as well as the ICH, the imaging-based signal generations. So therefore, they are quite a comprehensive brain cell atlas for the mouse and the marmoset and the macaques. So as a part of the Chinese brain project, the map of the macaque cerebral cortex was first published in Cell in the last year in the journal. So why did we only publish the cortical data? And first, you can see that this is a very large, massive project involving over 100 million cells, and with the raw data of 500 gigabytes. So the data for the other brain region is actually under analysis, with most of these already complete, and some of them have been submitted to the magazine for review. So we believe that the whole data set will be released to the public very soon. So this resource achieved has received widespread. So sorry, I will turn on my videos for. Okay, can you see my video? I'm sorry, so I missed the method.
Yes, we can see.
Okay, so the screen is okay.
Yes, you can go
Okay,
ahead.
okay. So what I want to say that actually this is a true team science effect. It says so involving the cooperation among the different institutes within the BGI, including the BGI resource, MGI, and the BGI genomics and so on, and also the as well as the extra partner from the, for example, for the Institute of the Neuroscience in Shanghai and the Tencent based in the Shenzhen, and so on. We have many people involved in this study. So this is the mouse brain mapping. So I will play the videos. So showcase the brain's cellular landscape in the details. So very different color also represent the cell types. So we can see the very details of the distribution of every single cell in the brain, as well as the gene expression for all the genes in every one single cells. So the origin colored is the olfactory bulb, and the lead colored is the hippocampus, and the outermost cortex layer are just a few examples of the many brain regions that can be distinguished in this whole brain atlas. And we also implement the DCS technology and construct our reference atlas, and which relieve the change in the hypothalamus of the macaque model of our diabetes and obesities. So while the INF is a very famous nucleus in the hypothalamus. So people always think that the INF nucleus was very commonly regarded with a sensitive region for the metabolism disorder. But our research have our interesting finding that besides the INF nucleus, and we also found the PVN nucleus and XKB showed the more significant change in the diabetes samples than in the obesity samples. So this novel discovery was made possible by the large datasets generated from the hypothalamus samples output. So this paper is also published in this year in the Cell Metabolisms. So next, apart from the well-known model species, and we are also interested to the evolutionary process of the complex organs like the brains, where we are very familiar with the species evolution through the DNA sequences, which allow us to reconstruct the phylogenetic tree for all the living species based on the Darwin's signals. So we believe that the organ, just like the brain, may also have the evolutionary features. The human brain, which is our complex the structure and the complex the functions, actually didn't suddenly appear, but rather evolved gradually over the hundreds of millions of years. So therefore, and study the brain evolution from evolutionary perspective is very important. So making the significant events such as the emergence of the neuron and the emergence of this central neural systems and the emergency of the spinal cord, and also in the end is emergency of the cortical nucleus. We call it the nucleus. So it's closely because the nucleus. Why people is interested in the nucleus? Because the nucleus is related with the advantage cognitive functions. So we firstly investigate the evolution of the cerebral cortex, which is playing an important for the, as I mentioned before, as the cognitive functions. However, we know that besides the mammals like human, birds also possess a remarkable high level of the cognitive abilities, which are widely believed to be the result of convergence evolutions. Interesting, when we combine the brains anatomy of the species between the human and the bird, so the brain region associated with this ability appears to be quite different. For in the mammals, and actually is the expansion of the new cortex is primary, while in the bird, and the expression of a region called the DVR and the dorsal ventricle region is more prominent. So in this project, and what is the homologue region between the birds and the mammal's brain is the first question for the study of the cortex, the evolution of the cortex, and the same for the hippocampus, and also for the layer structure in the neocortex in the mammals. But we can see in the mammals, actually we can see have the 6 layers in the neocortex. But in the totals, actually they only have three layers. But what are the homologue regions between these different layers? So it still remain poorly understood. So we firstly constructed the brain atlas of raptor, the Chinese soft shell toad, and aboard we named the zebrafish. So using the Stereo-seq, and we also including the dataset from published in the Cell, and also the Science I mentioned before, and so altogether, and we have 5 species in the Amniotes. I also include the outgroup, is amphibia named the axolotus. So the data analysis, and we discovered that by using the unsupervised clustering, and can effectively distinguish between different brain regions, as shown in the right side. And we can see more details than traditionally autonomy result. For example, in the totals, actually we can subdivide the cortical regions actually in this layer, and unlist in 4 distinct areas for the first time. So we can see, and this one, and this one, and this is hippocampus. So and also in the board, also we have observed laminar-like distribution of the DVR is very similar for the layer structures. So comparative analysis between the DVR and the neocortex, and our results show the significant difference in their similarity and in terms of the TF and effect genes. And we know that the TF are generally considered to be development and evolutionarily conserved, while the effect genes are more functionally related with the functionals. So this divergence suggests that the different structure and the structural evolution story can be identified by the data. So taking the bird and the mammals as example, and we can see that in the bird, part of the DVR region is named the independent regions. So is a very high similarity with the mammals neocortex but only in the effect genes. But when we come to the TF genes, actually the lead panel is similar with the other region, not the neocortex. So, but for the another region of the DVR, we named the hypopaneas. Both the effect gene and the TF genes have shown the similarity with the neocortex in the mammals. So this result indicates that only the leader paneas actually undergoing the convergence evolutions in not the whole DVR have been traditionally mentioned. So therefore, and through the comparative analysis, we identified homology regions across all the board and the totals and the mammals. So we use the divergence of the T and the effect genes to explain the phenomena of the convergence evolution in cortical structure and functions. Furthermore, and we characterized the two different TF evolutionary models. We named the capital and the shift models. So the capital model indicated that the regulation and relationship between the TF and the downstream effect genes actually have been conserved across the species. So as they showed, the board and the totals are the same, and the co-expression in both species. But for the shift model only, and for their relationship between the TF and the effect gene have been changed a lot across the species. Because as the case in the NR2F2 as in the case in the board, and actually only in the board, this TF have been regulated the downstream effect gene like the FEZF2 and the CUX1, but in the totals, actually just effect gene have been actually didn't expressed in the total place. So and also we can use our method, the CMCS, to very easy to distinguish between these two models. But as we believe that then in the future refined distinguish between these two models, we need to be more data and more resource. So this is a part of the network between the TF and the effect genes across species. From which we could find that the COPT and the shift model in the network. So so this is another project very similar for the spatial one, but we only focus on the single cell data. So this is our single cell data including the 5 species in the Amniotes. So besides the species I mentioned in the spatial and the Stereo-seq project, and we also including the pigeons in this single cell data set. So by performing the GCN and the method and the gene family information together, and we can integrate the single cell data from across the species with our divergence times of over 300 million years. So it's very difficult to do this. And from this data, and we can easily to see identified the cell type, including the conserved cell type. For example, for most of the non-neurons, a cell actually is conserved, but also the divergence cell type, such as the most of the excited neurons, and also some cell type in the cerebrus is also have a very high divergences. For example, for the Golgi cell and the Purkinje cell, and also the Bogman cell in the cerebrus. So among the excited neural cell types, we observed our significant divergence between the board and mammals. So such as the near, so we can see in the board actually is a very traditional mark genes named the SLC17A7 and A6. This is two microgenes used in the mammals. So we can see the both actually didn't express the A7 ones, but mammals actually express both of them, but more about the A7s. So when we come to the gene mutations, so we can find that the A7 has undergone many mutations in the body, and some of the species even loss of the genes. For example, for the chicken and the pigeons and some other species. So this is the case of the case of why the gene mutation can reflect the cell type in the functional and in the brains. So we can find that the protein fold. This is a result from the large model to like the AlphaFold to do the protein folding and for the A6 and A7s. So in the earlier weight blades, and actually they only have one copy of the A6 we named it. But in the earlier of the Amniotes, and they have duplication mutations in these genes, and we have get our parallel genes we call the A6 and the A7s. So but in the subsequent evolutions, and the mammals and boars have undergone very total different paths. So in the mammals, both the A6 and the A7 have been retained, but when we look at the gene expression spatial patterns, the total difference. So for the A6 and the mammals expressed in the older cortex regions, but for the neocortex, the many expressed for the A7, but in the board and they are total difference. So only express the A6. So besides the brain of the amniotes, and we also get the three-dimensional brain map of the lamprey, because lamprey is very interesting because it is one of the oldest living lineage of all the vertebrates. So we also get the data to analyze the cerebellum, the emergency of the cerebellum, and also very, very important brain regions. Also, we were very, very fortunately enough to get a brain sample from the natural from a naturally strand dolphin and whalefish with a special case in mammals. We know that this is species like the dolphin and return to the marines life from the land, and their brain and other systems have undergone significant changed. For example, we know that the dolphin also have extremely stronger cognitive abilities, but also the brain size even bigger than the humans. So in this project, and when we compare it with the mammary mammals between other mammals, we can help us to understand the various important issues such as the brain function, plasticity, and also the sleep, because we know the dolphin have a very total different sleep models, and also the spatial localizations. So this is very interesting. So regarding our series of the study on the brain mapping, brain outlets, and we have organized about 23 papers, and have been submitted to Cell Journal series, and we are expected to be published as special issues, and the article covered very large, so covered from the for example, for the outlets and the evolution and the development, and also the disease, and also we have some method. So we, especially in the evolution and the evolutive study of the brains, and and also the method BGI have made a significant contribution to this field. So, however, and we also know that as the technology advanced, the data size is also growing very fast. So that's why the life science will become a big data science maybe in the futures. So for instance, if we want to we get the human brains with the 3 dimensions. So we estimate about we need to over 1 to 10 billion cells, and the data size should be over the 10 petabytes. So is amazing data size. So therefore, maybe we also noticed that the advantage of the AIs and the research have been using the AI technology into the single cell genomics data. So for example, for the gene formal, and is very I think it's a very breakthrough use the transformer to the single cells, and also the SCGPT and also cell to sentence. So it's a very amazing progress. Now, but we need to pay attention to the programs in the UCE, is published in the Bellcaf in the early of this year, and we think this model can be used in the future of the single cell and spatial data sets across species, because they use the protein large language to do the homologous genes finding. So based on the large language models, we can use for example, for the UCE and the Saturns. And actually, we are trying some interesting study in the across species, especially in the brains. But we know actually for the limitation of the large language, we know that the direction of the cell embedding is lasting. So it's lost in the direction. So we are thinking about, and we proposed a new concept. Why if we can construct an evolutionary development cell tree by integrating our biology knowledge, and we can construct and also imputate the limitation of the large language models, and provide a more comprehensive understanding of the cell evolution and the development. So, and also, the BGI have a vast amount of the single cell raw data, both published and unpublished, across different species, and is a continuous generation new data we are doing. So to date, we have collected about more than 19 million high quality single cell data, and for the single cell model, we based on large language, and this time we can try the model beyond the language and consider the development and the evolutionary relationship between the cell type. So in that we are trying to use the model that take into account the topology structures in the large language. So there's another motivation for we want to do the integration across the different platforms, as we know, and besides the BGI's ST-omics, and we also have some amazing technology based on the imaging. So for example, for the morphage, and also some other technology is the same. So why we can integrate the data together and to improve, because every technology have the advantage and the disadvantage. For example, for the image based, actually for the capture rate is very high, but for the sequence based technology, the capture rate is a low. But for the diffusion issues, the image based is very low, and but for the sequence have our diffusion issues. But for another, in contrast, and actually the image-based is a panel like sequencing technology, but for the sequencing we have get all the genes. So when we cannot combine these two technology together to imputation for the Stereo-seq, and also is the same for the morphage. So that's we want to do another thing is we named the spatially construct a closed platform integration of the spatial transcriptors. So it's also based on the deep learning network to do. And the last I just show the result, and this we based on the MNN pair and distinct to mapping these two sections in the same coordinations. So in the end, we can use the mapping per end to the source slice, and finally spatially construct the pair and mapping the sequence-based and the image-based PCA spatial together. And also we can embedding for the thing for the cell for the embedding spatial, also for the protein embedding, also for the spatial embedding spaces. So we can do the imputation, and also we can do the enhance the gene expression for all the platforms. So this is the result we just tried. Very crazy. We only show the about 3,000 pairs, but actually we could get the more than millions of the pairs. So we very detailed for identifying the single cell levels. So and thanks I want to thanks our colleagues in the BGI and also the Stoker, especially for the ST-omics the teams, and also we want to thank our cooperation in the Institute of the Neuroscience in Shanghai, calling the both teams, and although we want to some other cooperations. So thank you all, and this is the end of in my presentations. So I will come to the questions. I don't know how many times I still have to have the questions to answers.
Okay, Dr. Liu,
Okay, Dr. Kim.
thank you so much for your presentation.
So I want to make sure how many times I have the Q&A part.
Maybe just 2 or 3 questions is available.
Okay, I will go to the text.
I will present a question for you.
Yeah, there are four questions in the I can see, and the first one is are there any special considerations for brain science analysis that should be taken into account compared to other tissue samples? Yeah, and I think for besides the brain tissues, actually we also perform the technology in the other organs. For example, for the liver, and also for the lungs, and also for the immune systems. So it's very comparable to do the comparative analysis across the species. So it's very, I think it's no problem for our ST-omics for other tissues. Yeah, and we go to the second questions. Are there any specific consideration I should be aware of when isolate single brain tissues? So I think because for the single cell and for the ST-omics technology, and we actually the issue, the main issue is the samples, the quality of the samples. So we generally use the fresh flash sampler to do the Stereo-seq and single set data. So I think if you sample is fresh and we can frozen for the frozen into the use the ice and some other, and also we have the update version of the Stereo-seq for FOP, and also I think we could do in the FOP in the future, and maybe we have been have delivered in some of our KOL. So for your question, I suggest in the current in the currently stage we as we use the flash sample, we should be better. So we go to the next one.
Yes, you can.
Yeah, this is a bit of topic. In China, are there human cell alternase, cancer cell alternase project using single cell analysis technology be well candidation? So yes, and as I know, BGI and the other institute in China also the part of the human cell alternase and also the cancer cell alternase. So the single cell analysis technology surely is very actually we use the we also cooperate with our cooperation in the international, for example for the American in the HCS teams, and also in the European for the young model, and we also have cooperation with the so actually yes, we cooperate with the team of the HCA and also the cancer cell alternate, but mostly of the HCA we are very know each others. So the next, this is a very good question. Whether the ST omics can be applied to the plant, and actually in the first batch of the papers we published in the cell series, I remember it should be the development cells, and actually we performed the ST omics technology on our plant. So in my personal opinion, actually I think the plant tissue and the plant samples have been more available for the ST omics. You know, we have the cell boundary in the plant. So actually in the plant is more, we can get more signal from the ST omics than the animal cells, because the animal cells have the diffusion issues, but for the plant cells, actually we can very easy to find the boundary of every single cell. So how we have published. So the last
yes,
one.
and next. Yes, this is the last question.
Okay. So what are the most critical technology limitation challenge that you faced when study brain evolution and evolution and the moments with currently available tools? And what would be the future breakthrough that likely overcome those? Yeah, also you are very important questions. So I think in the evolutive study, because basically we need to do the comparative analysis across species. As you know, across species is very difficult to find the homologous gene cell types. So the biggest question for us is how to define the homologous cell type to across the species, even especially for the large scale of the project, even for example for the fish and for the mammals. So it's very high divergence. So I think, and as I mentioned in the end, and we are trying to some latest technology, for example, for the AI from the large language model. So based on these language models, and Actually, for example, for the UCE and the cytons have been very amazing program in the have been developed by the Stanford University teams, and they are very high quality between the homologous cell type finding in across species. So I think in the future, maybe we could, and if you have interest in, I encouraging you to do how to developing some programs to do the homologous cell type and homologous cell homologous spatial domain identification in the future, and also based on some of the large language or even the other big data technologies, we can I think we can do the multiple omics data integrated. So in the future, I think the data will be very huge. So if we can catch up with the AI technology, maybe we could do the individual projects will be more better than currently studied. Yeah, thank you.
Okay, Dr. Lee, due to the time constraints, we will wrap up the Q&A at this point. Is there anything you would like to add?
No, thanks.
Okay. 네, 오늘 준비된 발표는 여기까지입니다. 브레인 사이언스의 스파셜 트랜스크립토믹스를 적용한 예시로서 포유류 신피질과 조류의 DVR을 비교하여 종래의 진화 관계를 전사행자와 이펙터 유전자의 시각에서 확인하고 뇌에 대한 근본적인 이해를 도운 유익한 강의였습니다. 시간 관계상 Q&A는 여기까지 진행되며 남은 질문은 다시 보기 메일을 통해 답변 전달드리도록 하겠습니다.